Okay? And the lips are formed by what muscle? Do you remember?
Maxillaris.
Right. The lateral walls are made up of cheeks, which are supported by what muscles? Buccinators. Remember we call those the trumpeters muscles? Or they blow and suck. Okay? The roof of the oral cavity is formed by the palate. Now, the part of the palate that's underlaid by bone is called the hard palate. On the other hand, the deepest part of the palate does not have any underlying bone. It's called the soft palate. In fact, when the soft palate comes down in the back of the oral cavity like this, it forms what? What's that? Uvula. Okay? Of course, the purpose of the uvula is when swallowing to close off the nasopharynx. So food and beverage doesn't go up into the nose. And then the floor of the mouth is formed by the tongue. The tongue has both intrinsic and extrinsic skeletal muscle. Intrinsic means that it's fully contained within the tongue. On the other hand, an extrinsic muscle is one that originates outside the tongue and then inserts into it. For instance, if this is the hyoid bone here, and this is the tongue out here, then you have the muscle that originates from the hyoid and extends into the tongue. That would be an extrinsic muscle. Okay? The tongue's attached to the floor of the mouth by a lingual frenulum. A lingual frenulum. Hunter, you want to turn around and face the class? I want you just to open your mouth wide and touch your tongue to the roof of your mouth. Do you see her lingual frenulum? Okay. That serves a little bit higher. All the way to touch your tip to the roof of your mouth. Go ahead.
Oh, yeah.
She's not showing it like she did before.
It's the thing.
It's a little membrane.
Yeah.
You can get that.
Now she's sticking her tongue out, but not touching. Let's try it, Sarah. See if you can do it. See if you can demonstrate your lingual frenulum. Oh, sorry.
There
Do y'all
you
see
go.
it?
Perfect.
Very good. Okay.
Sorry about my coffee
I
back.
can't. I have a bad jaw.
That's okay. That's all right. So we said that the mouth is important in mastication. Okay. It mixes food with saliva. So when that food becomes lubricated, when it becomes mixed with the salivary enzymes and the water from the saliva and becomes more macerated, we call it a bolus. Okay? And then when we form a bolus, then we initiate swallowing. We push that bolus to the roof of the mouth and we initiate the swallowing reflex. If you look at the surface of the tongue, it has tiny microscopic projections. Okay, that look like this, look like little conical projections called papillae. Okay. So, because they're on the tongue, we can call them lingual papillae. These are little upward projections, conical projections of mucosa. Remember that the mouth is going to be surrounded by a non-keratinized stratified squamous epithelium. Okay. Now, the most abundant of these papillae are what are called filiform papillae. Filiform give the tongue a white coating because the tips of the papillae here consist of keratinized epithelium, which is different from the rest of the mouth. So the tips of these little papillae are made up of keratinized stratified squamous epithelium, giving it a white coating. The purpose of this keratin on the tips of the filiform papillae is to grip stuff, is to allow for friction, so that we can move the bolus from one location to another, from point A to point B. So that as we chew, we can move the food between our teeth, for instance. So filiform papillae, which are the most abundant of the papillae, they don't have any taste buds on them. So if you look at the surface of the tongue, most of what you see are filiform papillae. But you're going to see an occasional little red spot. And that little red spot on the surface of the tongue is formed by what are called fungiform papillae. these have the shape of a mushroom like this instead of a cone. Okay? And the reason they appear red is there's a central core of vascular tissue that goes all the way up to the surface like this. Okay? And that vascular core gives it a reddish hue. Now, along the lateral aspect of these fungiform papillae, you may see taste buds that look like little footballs. Okay. Containing specialized cells for taste. Now, what separates the posterior third of the tongue from the anterior 2/3 is this little groove. Okay, so the groove separates the anterior 2/3 from the posterior third of the tongue. And this groove is called the sulcus terminalis. The sulcus terminalis. Sitting in a row, just anterior to the sulcus terminalis, is a group of larger larger papillae called circumvallate papillae. Circumvallate papillae look a lot like fungiform, kind of mushroom in shape, and they tend to have the most abundant taste buds. Compared to any of the other papillae, they have the most abundant number of taste buds.
Why does the book just call them the valley papillae?
Because some books call them vallate papillae. It's kind of an abbreviated form of circumvallate.
Okay.
Now, behind the sulcus terminalis are going to be your lingual tonsils. These tonsils are not surrounded by a connective tissue capsule, so they're not considered a lymph organ, but rather lymph tissue.
Circumvallate is all one word.
Circumvallate is all one word.
Okay.
Okay. Now, all of these tissues that surround the oral cavity, whether it be the palate, the cheeks, okay, the tongue, all of these tissues possess minor salivary glands. Minor salivary glands open directly into the oral cavity. In addition to these minor salivary glands that are present within the tissues that surround the oral cavity, you also have major, or they're sometimes called extrinsic salivary glands. They're located outside the oral cavity, but they have ducts that carry their secretions into the oral cavity. For instance, the largest of the extrinsic salivary glands here is the parotid gland. It's located just anterior to the ear. Notice it's got a large duct. It's called the parotid duct. That's going to pierce the buccinator muscle to enter into the oral cavity. And it's going to empty next to the second upper molar on either side. This is the largest of the salivary glands, the parotid gland. What you need to know about the parotid gland is, as the facial nerve exits the stylomastoid foramen, it passes directly into the parotid gland where it begins to branch like this. And its branches provide innervation to the muscles of facial expression. Okay? So that if you operate on the parotid gland, you have to be very, very careful not to clip branches of the facial nerve, because it will leave the patient with ptosis if you do so. Now, located deep to the mandible, this can either be called the submandibular gland, or some people call it the submaxillary gland. Let's call it submandibular, but simply be aware that some books call it the submaxillary gland. Its secretions are carried by a duct that empties into the oral cavity on either side of the lingual frenulum. So there's going to be a duct here that runs to the undersurface of the tongue, emptying on either side, the right or the left, of the lingual frenulum. So again, that's an extrinsic salivary gland. It's got a duct that carries its secretions. And then finally, underneath the tongue, if you look here, these are sublingual glands. They produce secretions that are carried by about 10 or 12 different tiny ducts to the floor of the mouth. Now, these extrinsic salivary glands, these are organs. They're surrounded by a connective tissue capsule. And inside of each of these organs is individual glands that can be classified as either serous glands or mucus glands. Serous glands are glands that produce watery secretions that are rich in enzymes. Now, for the salivary glands, the major enzyme produced is salivary amylase. Right. Salivary amylase, A-M-Y-L-A-S-E. So again, serous glands produce aqueous secretions, watery secretions that are rich in salivary amylase. Serous glands, when you look under the microscope, using the typical H&E stain that we use, are going to stain darker, kind of a purplish color. Okay? We say that they are basophilic, or dark staining. Of a purple color. And if you look at the individual glands, in a serous gland, they tend to have a nice, round, centrally located nucleus like this. Okay? And again, the cells themselves stain a little bit darker, purple color. Now, the other type of glands that can be present within these extrinsic structures are called mucus glands. Mucus glands have more of a thick secretion whose purpose is more for lubrication. They don't contain enzymes. They are lighter staining, typically a light staining pink. We say they are eosinophilic. They stain light, kind of light pink. If you look at them under the microscope, the nuclei of these structures more flattened toward the basal surface of the cells like this. That's a lighter staining gland than this one here. Okay. So here's the deal. Parotid glands are made up almost 100% of the serous secreting type. Serous secreting units. By the way, all three of these glands are made up of, these grape-like clusters are called acinar glands, A-C-I-N-A-R. An acinar gland is one in which the secretory unit is shaped like a grape. So an acinar gland is made up of grape-like clusters like this. And when you cut through the gland, you're going to see a bunch of these little subunits, these little secretory units that are shaped like little sphere. So the parotid gland is made up of serous type acinar units.
Can you spell acinar again?
A-C-I-N-A-R.
Thank you.
N-A-R or E-R?
A-R.
Acinar. Whereas the sublingual glands are almost completely mucus secreting acinar units. And then finally, submandibular glands are mixed. you see both serous and mucus secretory units. Okay. So again, these glands produce either serous secretions or mucus secretions that help to mix with the food and form a bolus. Now, the salivary amylase that's produced by parotid glands and some mandibular glands are going to begin the process of carbohydrate digestion. So amylase acts on carbohydrates. It breaks the bond between the individual monosaccharides. Remember, carbohydrate is formed of a bunch of monosaccharides that are linked by covalent bonds. So you might have a big carbohydrate like a starch or glycogen, and what it's going to do is it's going to begin the process of breaking some of these bonds. Okay? So converting a larger carbohydrate into smaller polysaccharide fragments. So the process of carbohydrate digestion begins within the mouth through the activity of salivary amylase. It uses water to break some of these bonds, these covalent bonds between subunits. So you're breaking down a large molecule into smaller fragments, polysaccharide fragments. Next, we're going to take that bolus with some partially digested carbohydrates, but it's also going to contain proteins and lipids, other nutrients, and we're going to pass it down into the stomach. The organ that's responsible for the movement from the mouth into the stomach is the esophagus. Okay? The esophagus itself has no digestive activity. If you looked at those glands in the submucosa, those were light-staining glands, those were mucus glands that simply produced a lubricant to help glide that bolus down into the stomach. Okay? So the esophagus has no digestive activity, it's merely a passageway. It begins at the cricoid cartilage, passes through an opening in the esophagus. Okay?